Almost to some term. I'm not teaching. I'm not teaching. I'm not teaching. That's what Lilo's is at. It's Dr. Rob Roll. He teaches basic micro and human systems. I have no idea. I haven't seen him today. When I see him, I'll ask him. I have no idea. I'm not teaching anything summer term. They asked and I politely declined. Actually, no, it wasn't. I said no. I'm like, we're just gonna do that. Alright. So, honestly, this stuff for the most part is a lot of definitions. Or not a lot of definitions, but it's more terms than anything. Bacteria, a lot of fungi are decomposers, so they are involved in nutrient cycles. So they take organic material and they break it down into its components so other organisms can use it. So they play large roles in the carbon cycle, nitrogen cycle, phosphorus cycle, things like that. We'll talk a little bit about some of those. Our immune system, you know, as we went throughout this course, has an interaction with your normal flora. So when you're really, really young, your normal flora actually, to an extent, helps train your immune response. Because you have all that bacteria in places like the gut, that helps with the immune response. And also, those organisms that you have in places like your gastrointestinal tract, they interact with each other as well. So, I'm not talking about this anymore. Does anybody have an idea of what, besides your immune system, keeps the bacteria in your gastrointestinal tract from not just exploding in numbers? Like, why, if you have a bunch of E. coli and E. coli divides over 20 minutes, why aren't you like squirting E. coli out of your ears? Like, what keeps that?
Shedding.
So shedding, yeah, that's one of them. And immune response. Think about what else, depending on where you're at in the gastrointestinal tract, pH. Think about what other microbiology, organisms. Think about what other things are part of your normal flora, like what other organisms are down there. Like what would keep the number of bacteria controlled? Other commensals. There's viruses and things in there too. So you have bacteriophage and things in your gut, they infect those bacterial cells and they help keep them a certain number too. So between those bacteriophage infections and your immune response and all these other things that we're talking about, and the constant shedding, that keeps the E. coli from squirting out of your ears. It keeps the number kind of at a normal, I shouldn't say normal, but at a certain level, a certain basal level. So those normal flora are kind of like a little ecosystem in themselves. Critical components of all ecosystems. No pure cultures in nature. So like the stuff we work with in here, like you have a culture of Pseudomonas or E. coli or whatever, nothing's like that in nature, everything's mixed. And even on the human body, it's like that too for the most part. This is why if you're trying to collect a urine specimen from somebody, you have to do like a midstream test. They have to pee a little bit. That gets rid of all the bacteria that's like on the floor on their skin, around the urethral opening. That way you don't have a urine specimen where you're looking to see if somebody has an E. coli urinary tract infection. You have a bunch of normal flora in there because it's like trying to find a needle in a haystack. You can't figure out what's there because you have a bunch of normal flora contaminating whatever is causing the infection. In nature, you also tend to find these organisms in biofilms, and the biofilms can be very complex and usually in direct contact with like atmospheric, like oxygen and nitrogen and everything, you find aerobes. And underneath that, you usually find like facultative anaerobes, and underneath them, sometimes you find anaerobes. The facultative anaerobes kind of act as a bridge, a lot of times, between the aerobes and the anaerobes. And sometimes they can use things from the aerobes and anaerobes to do different metabolic processes. And they act as a bridge between the aerobes and anaerobes. Again, the major kind of cycles that the microorganisms ecologically seem to impact the most are carbon, nitrogen, and phosphorus. Those cycles all tend to be linked because what do you need to make a protein? Like what do you need to make amino acids? Nitrogen, carbon, not necessarily necessarily phosphorus, but you need nitrogen and carbon. What about to make like a nucleic acid, like a phosphodiester bond between nucleotides? You need all of them. Yeah, carbon, nitrogen, phosphate, you need all of those things. So that's why those cycles tend to be linked together. So they have all these microorganisms to actually survive. A lot of times tend to work together. They can communicate with each other through things like form sensing, where they can tell how many other bacteria are in that environment, and it allows them to keep their numbers at a certain level. That way they don't essentially starve themselves out. No microorganism has all the genes to break down all the different kinds of things you find in the environment. There's some microorganisms that have some really weird genes to digest a lot of odd things that you wouldn't think they're able to do, or survive exposure to different things. There's different microorganisms that come across that have resistance to arsenic and copper and lead and radioactive compounds and all kinds of stuff. But there's not really just one bacteria that is resistant to everything and can metabolize everything. Usually certain bacteria can metabolize one thing, their byproducts can be used by another organism, they can digest other things, and they kind of share metabolites, byproducts. But not one of them can do everything. Again, the bridge between aerobic and anaerobic bacteria tends to be facultative anaerobes because they can live in both environments and they act as a bridge between those two kinds of oxygen tolerant organisms. Again, in the environment, most of the microorganisms are going to live in some kind of a biofilm. They can make different cellular products. One of the cellular products, especially bacteria make, to actually start to lay down the biofilm as they release DNA, extracellular DNA. Sometimes you see that referred to as eDNA. Essentially, they secrete pieces and parts of DNA, and that DNA, I don't know what y'all do in some other classes sometimes. Does anybody ever like extract a bunch of DNA? I mean, like usually like what they do in like a high school class or something is they'll take like a banana and they'll extract on banana DNA. Yeah, it makes like, what's that DNA look like? Like what's the consistency of that? It's like really goopy, yeah. So it's like really goopy, sticky. Well, if you release pieces of parts of like fragments of DNA, it's goopy and sticky. It allows the bacteria to stick to surfaces. And as they stick to the surfaces, they start to mature into a bigger biofilm. So microbial kind of community. So that's kind of how that works. Again, these biofilms can have major roles in biogeochemical cycling. They can be associated with disease. We talked about that a little bit here and there. The real easy example, to understand for that is like somebody has a knee replacement. They have a knee replacement. No matter how good the surgeon is or how clean the operating room is, oftentimes what happens, you make a decision, you replace the kneecap, but what ends up happening is someone going forward from the patient's skin gets into that opening, and then they end up getting a biofilm on that knee replacement. Once you get that, those biofilms are resistant to your immune response a lot, and they're also resistant to different antibiotics, so really the only way to fix that is to cut them open again, take out the knee, put in like a joint spacer that has the antimicrobials impregnated into it, and treat them with antibiotics, and then go back and do that surgery again and put a new knee in. Because once it's there, it's not coming off, and it's very hard to treat. So that's kind of one of the examples of how these biofilms play a role in disease. Another example of that, and I think we might talk about this a little bit too, is sometimes you'll see people that use like IV drugs. Sometimes they'll be contaminated with different environmental fungi and things like that. And oftentimes it ends up going and attacking the heart valve. And when you look at the heart as it works, when the valves open and close, there's like a fungal balloon that kind of waves with to cause some really nasty infections to the heart too. So those can ultimately result in bad fluid. A lot of these organisms, especially in like streams and things like that, they're there, and things like rocks in the stream, and they detoxify things. Swamps are a good example of this. You have a lot of microorganisms in swamps. When you start to take away all the swamps, you tend to get less clean water in the stream surrounding the swamps, because the swamps oftentimes with all these microorganisms act as like a big biological filter, and they filter down a lot of the contaminants. So you take the swamp away, you don't have that natural kind of filtration system. What a simple biofilm looks like up there at the top, which doesn't really exist in nature ever. It's just one kind of microorganism and one layer across some kind of a substrate. That's not really a thing. The second kind of example is a little schematic of what I was kind of explaining to you. Again, you ever see a biofilm this nice and neat and it looks just like this? No. But in this example for the biofilm, you have photoautotropes on the front or on the top. So things maybe that are like photosynthetic, so that should make sense because they're up towards the top where you get light. And then underneath them, you might have some facultative anaerobes. And then underneath those, you might have some anaerobes. So again, facultative anaerobes are kind of the bridge between the aerobes and the anaerobes on the bottom. They never look like that. Usually they look like this. So in this kind of example, you'd have the anaerobes in the middle, some facultatives surrounding that, and then the aerobes on the outside. This is usually what a complex kind of biofilm in nature looks like. Nothing looks like that. Nothing's ever that perfect in nature. But this is a little bit more of what it looks like. Okay, so here's some of the terms. So cycling carbon requires some different kinds of processes. Mineralization, complete decomposition of organic material to a simpler organic compound. So say glucose, and they break it down into like methane or carbon dioxide or ammonia or something like that. So they take something complex that's organic and they break it down into something simpler that's inorganic. Versus fixation. So that's when you take a gas and you turn it into a solid, ultimately. So carbon or nitrogen change from gaseous to a solid state, and then it's used to generate biomass. So carbon fixation, different kinds of dark reactions. Take carbon dioxide and you fix it into something else that's solid. So gas to solid is fixation. How these compounds, these carbon compounds are broken down by different microorganisms, that depends on the kind of compound you're talking about. So the more highly branched the compounds are, the carbon compounds, the harder it is to break them down due to steric hindrance. So what does that mean? It means if you have something that looks like this versus something that looks like you tell it's been a while since they drew chemical structures on the board? Good Lord. If you have something simple, it's easy for an enzyme to come in here and cut something because the sites are accessible, okay? But over here, that's a little bit harder for an enzyme to actually degrade because how are you going to access maybe this point? You have a lot of things going on here and the enzyme might not actually be able to cut in that site. So that's what number one is talking about. So something that's highly branched, more complex, might be steric hindrance. The enzyme might not have easy access to whatever site it needs to access to catabolize. Over here it'd be easy. Cool? All right. Actually, I did myself a favor here. Many different types of linkages. Same kind of bonds, some more simple. Bunch of different things, different kinds of bonds, different things going on over here potentially. So different types of linkages means the organism might not make an enzyme that recognizes whatever that bond is, so it can't break that bond. It might contain metals or different halogens, so things like fluorine. And what happens in those situations, essentially when you take that compound, let's say bacteria uses an enzyme to break that compound, it might liberate a heavy metal, like a lead or copper or something. Gallium. Bacteria don't like that necessarily. So in actually breaking down the compound and degrading it, they're releasing something that's toxic to themselves, and by breaking it down, they essentially poison themselves. So that's what that means. So So a lot of different halogens, different heavy metals have toxicities to microorganisms. Co-metabolism happens sometimes too, so what does that mean? It means that the microorganism has an enzyme that, say, breaks down some kind of a sugar normally, but there's some other compound that it comes across that has a similar structure to that sugar or similar chemical bonds to that sugar, and it just by chance can degrade that compound too. So different examples of that, DDT, so some bacteria can break down DDT, it takes about 30 years. DDT is an insecticide. There's another compound that has a similar structure called 2,4-D. 2,4-D is a broadleaf weed herbicide. So, 4 words to actually tell you. You spray it on it, things that have big leaves die. That's essentially what 2,4-D is. And it takes about 6 or 7 days to break down that compound using the same enzyme that it can use to break down DDT that takes 30 years. Just because there's a little bit of a difference between those. Are there bacteria or fungi or whatever that just happen to make these enzymes to break down insecticides and herbicides? No, they use that enzyme to break down something else naturally. And we have so much pollution in some of these environments from these different herbicides and insecticides, they just come across that too, and they can use those same enzymes to break those things down just by chance. A lot of these times they won't degrade, like they'll degrade the compound, but they won't really get any energy out of it. So like again, say they're using this enzyme to break down DDT and 2,4-D. It's something they usually use as an enzyme in some kind of metabolic process. They usually generate ATP. Well, when breaking these down, that enzyme might break these down, but they don't get any ATP out of doing that. It just recognizes bonds and those things too. There's a lot of examples of this when you do stuff environmentally. There are different bacteria we know. If there's an oil spill, you can release the bacteria into that water source, and it'll actually chew up the hydrocarbons in the oil. What you usually do with those bacteria is you give them some kind of a deficit in being able to acquire, usually it's a metal, like you mutate a gene they use to take up iron or something like that, so they can't take up iron on their own, like use a steraport or something like that. You add the bacteria where the oil spill is, they gobble up all the hydrocarbon, and then you don't want that bacteria or whatever it may be introduced into that ecosystem. You take iron away from that ecosystem, and then they all die because they can't get on the ground. So if you release them around the oil spill, you dump a bunch of like iron salt into the water too. Once they consume all the iron salt, and hopefully all the oil too, because they can't get iron on the ground. So that's kind of a thought process with some of this stuff. If there's no carbon present, then nitrogen will not be used, again, because these cycles are linked. If there's, in the same token, if there's no nitrogen present, then carbon will not be used. That's kind of the basis for that citrate test that we did here earlier on in the semester. Remember, that's the one where it has that citrate utilization system that changed the media from a green color to a blue color. So that's the citrate test, not the Simon citrate test. Sorry, a little misspelling there. 2 M's, not 1. Okay. So again, a lot of these cycles tend to be kind of linked to one another. So here in this situation, you have aerobes over here that are fixing carbon. So remember, carbon fixation, you're taking a gaseous carbon like carbon dioxide and fixing it into something like a sugar. So that's carbon fixation. So gas over to the solid. Same thing, anaerobes can do that too. Gas in this situation over to the solid. Over here, you can have mineralization. So that's when you're taking the organic material and you're breaking it down into something that's inorganic. So in this circumstance, you're taking organic carbon and you're breaking it down into carbon dioxide, something that's inorganic. So that's mineralization. And what ultimately happens here is different bacteria, like these methanotrophs that are aerobic, and these archaea methanogens that are anaerobic, can share byproducts of these different reactions. Oftentimes what you see in the middle is some kind of a facultative anaerobe that acts as a bridge, but ultimately if you didn't have these, you couldn't have these. You didn't have these, you couldn't have these, because their cycles are linked in with one So if you eliminate one of these from the environment, the other one will automatically die off. Nitrogen cycles require to make amino acids, so amino acids are used in proteins. But again, since the carbon cycle and the nitrogen cycle were linked, you need the carbon cycle to be working too. No carbon, no nitrogen, no carbon. For the nitrogen cycle, nitrogen fixation is like carbon fixation. You're going from gas to solids, and in this instance, you're going from gaseous nitrogen to something like ammonia, so gas to solid. Some bacteria have nitrogenase complexes, and they're responsible for this fixation step. So the nitrogenase is the enzyme that's responsible for that nitrogen fixation. We will talk about this a little bit more in a little bit. That nitrogenase actually requires an anaerobic environment for that enzyme to function. So you find these organisms typically living in an anaerobic environment. So we will talk about that here now. So this is a unique kind of relationship. So you have nitrogen fixation carried out by microorganisms that have this nitrogenase complex. So what they do is some of these organisms that have this nitrogenase complex, like rhizobium, they'll take nitrogen from the atmosphere, like nitrogen that's in the room here at like 72%? No. Whatever. 70-some%, I don't remember. 70-some% nitrogen, take it from the air and actually convert that into a compound the plant can use for nitrogen to make things like amino acids. With legumes, they have rhizobium associated with the root system. They can convert due to nitrogen fixation, gas to solid. The plant, so even though the rhizobium is oftentimes associated with the roots of plants, it's not so deep down in the dirt that they don't get any kind of oxygen exposure. Sometimes you see rhizobium associated with nodules that are actually above the soil with these legumes. Remember, for that nitrogenase to actually do nitrogen fixation, it has not to be exposed to oxygen, or the nitrogenase doesn't work. So what these legumes do is they make something called leg hemoglobin. The leg hemoglobin actually binds up oxygen. So it makes the environment oxygen free for the nitrogenase to convert the gaseous nitrogen to the nitrogen that plants can use to make things like amino acids. So it's this kind of symbiotic relationship. If the plant didn't make the hemoglobin, that rhizobium wouldn't be able to fix nitrogen for the plant. Get nitrogen. I feel like I went around like a mile around that. Does that make sense? So there's a symbiotic relationship. Plant needs to actually make the leg hemoglobin for the nitrogenase and the rhizobium to work. This is a very basic nitrogen cycle. Again, some more terminology. You can have things like nitrogen fixation, which we talked about, gas to solid, and then you can have nitrification. So where you're going from NO2 to NO3, that's nitrification. It's an oxidation process. This is usually carried out in an aerobic environment. And then you can also go from things like NO3 up to gaseous nitrogen, and that process is called denitrification. So that's a reductive process. And you usually see denitrification carried out by anaerobic bacteria. And then these different compounds are fixed into amino acids. The process by which those nitrogen compounds are converted into amino acids, or actually incorporated into amino acids, you should say, it's called assimilation. So specifically in this case, it would be nitrogen assimilation. So again, there's a lot of this kind of terminology in this one. So know the difference between nitrogen fixation versus nitrification versus denitrification. Just a lot of terms. We will talk about exam 5 here in a little bit, but pretty much the only question on that exam for that applied environmental micro lecture is this first bullet point, because I talked about that in that lecture too. Microorganisms can change the toxicity of metals. Essentially, for these microorganisms. They can add or subtract electrons to change the oxidation states of metals, and they can also add functional groups onto the metals that make them no longer toxic to said microorganism. This is the same thing that holds true for antibiotics. If you have an antibiotic and you add some kind of a functional group onto it, like a carboxyl group or a hydroxyl group or something like that, it changes the functionality of the antibiotic. It might make the antibiotic work better. It might make the antibiotic not work at all. But changing those functional groups changes the function of that compound. For metals, for antibiotics, or whatever it is, that's pretty much the only thing you're supposed to get out of the environmental microbiology lecture that you get. If you stick different functional groups onto the compound, essentially changing what the compound is, that compound's going to act different because it's not the same compound anymore. Pretty much all you're supposed to get out of that. You find disulfide vibrio in sediments, and an example of how you can have functional groups on the heavy metals. If it's exposed to mercury, what it does is it methylates the mercury. And when it methylates the mercury, methylated mercury is very volatile. So what's volatile mean in this case? It doesn't mean that it makes it like flammable. What it means in this context, when you're talking about biological systems, if you take something like a heavy metal and you add some kind of functional group onto it, in this case it was methylated, what it does is it makes it volatile, which means it crosses cell membranes easier. So an example of this, if you have mercury versus methylated mercury is going to cross your blood-brain barrier and across the placenta a lot easier than just mercury. So it becomes volatilized, so it can go through cell membranes and different kinds of tissues easier. If it is double methylated, which means this has two methyl groups on it, it is even more volatile, which means it can cross the blood-brain barrier placenta even easier than the single methylated mercury. Real complicated, right? So that's these kind of compounds can concentrate in different animal tissue, resulting in something called biomagnification. And if you talked about that in other classes, especially if you had an ecology class, what that ultimately means is you have something low down the food chain that takes in, like, I'll give you an example, like lead or mercury, one would be good examples. That organism is, say, eaten by a fish. That fish is going to eat a bunch of those small organisms that each have different mercury or lead types in it. You get more of that mercury or lead in the small fish. Small fish is eaten by a bigger fish, it's consumed a bunch of those small fish. Then they have more of that metal in them, and then a bigger fish eats them, and then us being the biggest fish, we eat them, and then we get a huge dose of whatever that compound is. Lead, mercury, whatever. This was always a big problem up in Lake Erie because they would always tell us not to eat more than like 3. Forgive me, I don't remember the fish that are in Lake Erie anymore. But they would tell us to only eat so many of those fish like every month, because there's a chance if you ate more than that, you would be essentially giving yourself like lead poisoning, because you're eating a bunch of lead that's biomagnified in those fish. Exactly what we talked about. Okay, so again, some more terms. So the different kinds of microbial relationships you can have mutualism, and we'll walk through all these. Commensalism, which is kind of what skin microbes are with. Commensalism, you're usually seeing an interaction where one organism benefits and the other organism doesn't really seem to get anything out of it. Predation, obviously one of the bad relationships. Predation is where you have one organism that's actually killing the other organism. Parasitism is where you have one organism that is actually somehow getting some kind of a resource from another organism. And as they do that, they are essentially parasitizing that other organism. They're not necessarily killing that organism, that's predation, but they are essentially extracting resources from them. And then you have mensalism, and we'll talk a little bit about that here shortly too. That's when some of these microorganisms produce a compound that kills or controls other microorganisms. Different examples of that, things like fungi that make a bunch of different antibiotics. You see those in the environment. They kill off other things that live in the environment with them so that they have more food for themselves. So that's mensalism, making some kind of a compound that controls the growth of other microorganisms. So let's talk about this one first. So you have mutualism into symbiosis, so both organisms are going to benefit. Sometimes you see endosymbiotic kind of couples. I can talk about this for people too. I'll give you these examples since they're up there as well. But you have microorganisms in your gastrointestinal tract, things like E. coli. Remember way back towards the beginning of this course, what did I tell you E. coli makes that you can't make in your gut? Anybody remember that stuff? Vitamin K, makes a bunch of B vitamins too, B6, B9, B12. Makes a bunch of B vitamins, but it makes vitamin K. So vitamin K is involved in clotting. You can't make those B vitamins, you can't make vitamin K, your body doesn't have the ability to make those things. So if you don't have those E. coli in your gut to make those things, you get a cut, that cut's not going to stop bleeding because you can't clot. So those E. coli are in a symbiotic relationship with you because they get a constant influx of food to feed them when you eat, but they're also providing B vitamins and vitamin K to you as well. So symbiotic benefit. Same thing holds true for cows. Rumen breaks down cellulose because of microorganisms in the cow rumen that breaks down cellulose. And then the cow receives more nutrients from that broken down cellulose sugar. And then also those organisms in the cow rumen keep on getting food as the cow eats. Another example, squid carry bacteria and light organs that generate light for the feeding and camouflage. So they generate light that attracts things for the squid to eat, and then they get more food, and the squid gets food too, by then making light. Another example here, sulfide-based ecosystem at the bottom of the ocean. You know this one. This worm has hemoglobin that bites on the hydrogen sulfide. That hydrogen sulfide is oxidized and used to make ATP, and then the ATP is used to make sugary carbon compounds. The worm gets more nutrition, and the microbes have a constant source of nutrition coming in as the worm feeds. So again, both organisms are going to benefit. We talked about this, both organisms are going to benefit. Same thing here, just showing you what it looks like. The squid as well as the microorganism that's actually producing the light, they're both going to benefit from this interaction. Alright, so let's talk about this a little bit. So there's a special kind of mutualism called syntrophism. So that's when the metabolism of one system makes the energetics of the metabolism of another system favorable. So we kind of talked about this a little bit. Essentially, down here in this relationship, you have syntrophomonas, makes things like hydrogen that the methanogen needs to make methane. Without the syntrophomonas, the methanogen doesn't have the hydrogen to make the methane. So you have to have both of them. Without one, you can't have the other. Some different examples of this are cross-feeding. So that's when one organism gives the organism something else, to make something for themselves. The example here for syntrophomonas. For syntrophomonas to actually grow well, it has to find somewhere to give those hydrogens to. Because if it gets too high a concentration, it's bad for the syntrophomonas' growth. I was just double checking to see if I actually put this up on this slide. I guess I didn't, so I explained it to you. So both of the organisms here benefit from this because the methanogen is taking that hydrogen away from the syntrophomonas so it can grow well. So that benefits the syntrophomonas. But the methanogen without the hydrogen isn't going to be able to be in a methanogen. So they benefit one another. Sometimes you see that referred to as cross-feeding. One organism is supplying some kind of nutrient or something to another organism. So they're cross-feeding. Form sensing, we've talked about a ton of times in here, so I'm not going to bother with that again. But essentially, you can tell the number of bacteria, you can tell the number of bacterial cells in that environment. Commensalism, so one organism benefits and other is neither harm nor help. Again, when we talk about that with normal flora for humans, how many of these, say, skin flora actually don't benefit, like we don't benefit from them being there, or we just don't know how we benefit from them yet? Probably most likely we just don't know how we benefit from them. So we call it commensalism because we know they benefit, but we're not entirely certain how. We know how they would fit, not really certain how we would. So one organism gets something out of it, another organism doesn't. An example of this, waste brought to one organism serves as a substrate for another. So nitrosomonas and nitrobacter do this where they have to kind of live with one another for nitrification. So they can, the nitrobacter takes the waste product from the nitrosomonas to actually survive. So that's a kind of a relationship. Do we talk about E. coli abdominal infections in here at all? So remember, if I did talk about that, remember I said sometimes you see somebody gets in a car wreck or something and like a gut perforation, they'll get E. coli out of their gastrointestinal tract, like their abdominal cavity. And I said that you always find E. coli with Bacteroides. This is the reason why you always find E. coli with Bacteroides. So Bacteroides needs an anaerobic environment. E. coli aids in creating that anaerobic environment for them. Therefore, you always find E. coli associated with Bacteroides. So if you have somebody that has a Bacteroides infection in the gut, E. coli is there with them. Always. They're always together. Another example of commensalism is Vibrio cholerae and chitin on different kinds of crustaceans. What that Vibrio cholerae does is it makes a chitinase that aids crustacean eggs in actually hatching. However, those crustacean eggs will hatch whether you have Vibrio cholerae or not. So it's kind of one benefits. The crustacean really doesn't the eggs will hatch without it. But what can happen is the Vibrio cholerae can take the chitin, break it down, and so breaks it down to make ATP. So it benefits well. The crustacean doesn't really necessarily need the help. So that's commensalism too. And then we talk some more about these kinds of things. You have microbial predation. One organism kills the other organism. Padella vibrio preys upon E. coli. Sometimes you see those referred to. Padella vibrio is a bacterium. You sometimes see referred to as lifting antibiotics. They prey upon E. coli and E. coli cells, kill E. coli cells. So that's predation. One organism kills the other. We talked about this at the beginning of this little session here. Phage infects and lyses E. coli. That's one of the ways E. coli populations are controlled in your gut. There's phages there too. The phages lyse the E. coli cells. When those organisms are predated upon, nutrients are released back into the environment so other microorganisms can use them. And then you have parasitism, so one organism is going to be harmed, but not necessarily killed. That's predation. So one organism gains and the other one's harmed somehow. Different examples of this, you have EBV and lymphocytes, so Epstein-Barr virus. It's there, it's hanging out. Nothing really comes of that though, they're in latency. Sometimes that is kind of stabilized, that relationship stabilized over time. Remember I told you that a lot of these microorganisms are super duper lazy. So as they get in an environment where they don't need to work very hard to survive, they start to get rid of genes they don't need. I gave you an example of this many classes ago now with endogenous retroviruses. I don't know if you all remember this one or not, but over time we've had this virus that's kind of integrated its genetic material into us. And if you get somebody that's pregnant, and they did these experiments with sheep, if you take a sheep that's pregnant and you take out the genes that are related to endogenous retroviruses, the placenta doesn't develop all the way. And so over time, there's been a genomic reduction and it got lazy, essentially to the point where the virus doesn't even exist anymore, only in just certain genes that's incorporated into our ribosomes over time. If you take those genes out, it results in miscarriage in sheep. So that is a parasitism relationship where it's become very simplistic over time. Other examples that we've talked a lot more about, coexistence between the host and the commensal can shift to the pathogenic relationship. So if you have, say, some kind of something that's a bacteria that lives on your skin normally, it's normal flora, normally it's there, it's protecting you from other microorganisms coming in that might be a pathogen, causes some kind of skin or soft tissue infection. but then you get a cut. And then that microorganism that's normal flora and usually not causing any problems, causes an infection in that cut, like say it's Staph aureus or something. So that opportunistic pathogen is meant from a commensal where one organism benefited but the other one didn't, into something where, oh, the pathogens make you sick now. So it's more of a parasitism relationship, or parasitic relationship. And then you have, we talked a little about this already too, commensalism. Some of these organisms are going to produce antimicrobial compounds to control the growth of other microorganisms so they don't have to compete with them for food primarily. Negative impact of one organism on another based on release of some kind of compound. It's usually some kind of antimicrobial or an antibiotic by bacteria or fungus. Different bacteria make bacteriocin. Usually those are bacteria within the Bacillus genus. That's the main bacteria that makes bacteriocin. In the colon, bacteriocins protect us against some pathogens because they're present. If you get something in the gastrointestinal tract that might be pathogenic, those compounds secreted by your normal flora have the potential of killing off that pathogen. Does it always kill off those bacterial pathogens in your gut? No, you never have any kind of bacterial infections in your gut. That happens sometimes. But that's supposed to help keep the occurrence of that less. Another example of commensalism, leaf cutter ants. They create these fungal gardens to feed their young, and the parasitic fungi there are different parasitic fungi that can destroy the garden. But what happens is they carry streptomyces on their exoskeleton, and that streptomyces, remember we talked about streptomyces? Remember streptomyces makes a whole ton of different antimicrobials, antifungals, and antibiotics. So as they carry that on their exoskeleton, it releases all those antimicrobials to help protect these fungal gardens they use for a food source. So that's another example of commensalism. For us, we make defensins from epithelial cells and Peyer's patches that line the intestinal tract, and then they're released by us and different other species of eukaryotes, and it can impact what different kinds of pathogens that you might be susceptible to based on what the range is of the different bacteria and defenses you kill. Okay. So, long story short, microbes been around a long time. Anaerobes first, aerobes second. Gotta have them around. We don't know crap. There's tons of bacteria out there. You can go out into the ocean, you can look for different genes, and you can do ribosomal RNA sequencing. You can find all kinds of microorganisms that we know are there or have been there based on what you get back from sequencing. We don't know how to culture the vast majority of them, and ultimately for a lot of these, if you can't grow them, we can't figure out how they work. The example I gave you all in here of that is when you go to like say you have to take an antibiotic for something, and the doctor says, well, you need to take some kind of yogurt to kind of replenish your normal flora. And you look at the bacteria that's in the yogurt, and it's like some kind of lactobacillus or lactococcus and some kind of streptococcus. There's usually like 2 or 3 things in there. Well, your gut flora obviously is a lot more complex than that. Those are the 2 or 3 we know how to grow. The rest of them we don't know how to grow. It's really hard. So about 1 to 5% of all these microorganisms that exist we know things about. One of the main ones being Ecology, because it's kind of a model organism for better or worse for bacteriology. But the other 99 to 95% we don't know a lot about. We know maybe they're there, or maybe they were there, and that's about it. Yeah.
But it would you would still consider it beneficial to like consume that? Because like for
Yeah, it's very
nothing.
Yeah, absolutely. Yeah, I mean it's better that you're replenishing something, but it's just not. This is kind of goes back to the example we gave of Clostridium difficile. If you have a Clostridium difficile infection from like say you take an antibiotic, it kills off some of the other normal flora, the Clostridium outgrowth, you get that kind of infection. If you're trying to treat that bacterial Clostridium infection with antibiotics, like 95% of the time that fails. Because what happens, so you all know this now. So Clostridium, what's Clostridium? Clostridial species make? Bacillus and Clostridium. And make endospores. You give them antibiotics, And then when you take the antibiotics away, and the spores turn back into vegetative Clostridium difficile, and you got a Clostridium difficile infection. So what they call that is treatment failure. So this is where we talk about where you take a fecal transplant, they take normal flora from someone else, they freeze dry it, they put it in the capsule, you swallow it, and then you get that person's gut flora. That works because you got all the bacteria in that capsule from that freeze-dried food. We didn't grow that. We took some poop and we dried it out and put it in a capsule. So we didn't have to know how to grow it. I mean, in theory, if you could grow up all those bacteria, and you knew how to grow all them up, you could you could grow them all. But we don't know how to do it. Okay. Turn in your lab. I'm trying to make sure I don't forget anything.